In this video lecture module, we'll be talking about embryological development. So in our last video, we were talking about this picture here, showing about 10 days after implantation, how the developing embryo has surrounded itself with this green layer, the syncytiotrophoblast, and then this purple layer, the cytotrophoblast, and within the very first few layers of the embryo. Remember, this blue bubble is the amnion. The yellow bubble is the yolk sac. And this so-called trilaminar disc. Lamina means layers. So 1, the blue layer, 2, the red layer, and 3, the yellow layer. This is the trilaminar disc, and it will develop into the embryo. The process of creating those first 3 primary germ layers, in this sense germ doesn't mean infection or bacterium, germ meaning the seed, the stem cells that have the capacity to specialize into any of the hundreds of adult types of histological tissues. These are the layers we're going to see in just a moment. The ectoderm, the mesoderm, and the endoderm. Here's how it happens. Right before that, these 2 layers are called the epiblast, shown in blue, and the hypoblast, shown in yellow. This is the most complicated part of embryology that you need to know for this class. Here's how it goes. The epiblast cells start to undergo a lot of mitosis, pushing the cells towards the center and down through this little groove. Think of this groove as like a magical doorway. As the cells pass through the magical doorway, they change into these little red cells, the mesoderm. Once all of these epiblast cells have passed through the magic doorway, and we've made as many mesodermal cells as we're going to make, then the process stops and we change the name. The epiblast becomes the ectoderm. There are some internal morphological changes that are occurring with that name change. The hypoblast, which is this yellow layer surrounding the yolk sac, after this magical doorway process we call embryogenesis. After that process is complete, we change the hypoblast name to the endoderm. And again, there are some changes. You can see these cells actually replace the other cells through gene expression and differentiation. So just glance down again. We can see in context where the ectoderm, mesoderm, and endoderm are relative to these other fluid-filled cavities. So what? Who cares? Why do we need to know this? Well, this is really what you need to know. Down here on slide 69, to make a connection. After embryogenesis, organogenesis begins, and we see what happens. The ectoderm becomes the epidermis, the nervous system, the lens and the cornea of the eye, and the internal ear. You might remember from general A&P, which includes the cochlea, the vestibule, and the semicircular canals. The mesoderm becomes all of this stuff, and the endoderm here, bolded, probably important. Even though it might only be a question or 2, you should be prepared to recognize which adult tissues derive from these 3 layers. Okay, going back and adding just a little bit more detail, we see here just a few days after embryogenesis, the little developing embryo begins to fold. This is sort of a sagittal section on this column, and then maybe more of a coronal or frontal section on this one. So sort of looking back and forth, we might imagine what's happening here. So the trilaminar disc begins to form this primitive groove right here, the neural groove, which will become the central canal of the spinal cord and ultimately the ventricles within the brain. The blue tissue, the ectoderm, specializes, as we had just seen, to become part of the nervous system. Now, the reason I'm talking about it is this is oftentimes before a woman is even aware that she's pregnant. If she has had irregular cycles before, or she's not, really in touch with her monthly cycle or the calendar, she could be pregnant at this point and not know it. In the event that she has insufficient folate in her blood, the developing embryo has a very hard time forming this neural groove, and the baby could be born with a form of neural tube defect like spina bifida or anencephaly. So a little public health outreach here. Ladies, please take a multivitamin every day. Just for pennies a day, you can have the confidence that in the event that you do have an unintended pregnancy, that baby will have all of the nutrients that she needs for normal development. Okay, what happens next is sort of an elongation of the yolk sac, and as we had talked about in the prior video, the nutrients, primarily fatty acids and some proteins, cholesterol, good building blocks for a tiny creature. These are slowly absorbed into the primitive gut. You can see after that folding, we have created a little internal cavity. So as strange as it sounds, because this was originally the outside of the body, part of the yolk sac, the gut, even as an adult, is still histologically an external surface of the body, a strange tube that goes through the body but is not itself the inside, right? We'll talk more about that later on, but when you're digesting food, it's surrounded by your body, but still outside of it. Only when things are absorbed through the membrane and into these internal tissues do we really consider something to be on the inside of the body. Okay, we can see the developing head and tail here. By the end of week 5, the cavities for the lungs and the heart are beginning to develop. And actually back here around day 23, right here in the center of the mesoderm, we see the twitching of a primitive aorta, the beginning of the heartbeat. A couple of other quick things I want to point out on this video. Going back to this slide we had discussed briefly in the prior video, let's identify the different layers of tissue that surround the baby and separate the baby from the mother's body. Remember the name of this structure here, the interface, is the placenta. If we look here in more detail, we can see that although the maternal blood filling the placental sinus comes very close to the baby's blood flowing in and out through the umbilical cord, the blood never actually mixes. And we can see, focusing on this pink tissue here, we call the chorion. This is the membrane that prevents the maternal blood from actually mixing with the baby's blood. As strange as that sounds, the two blood systems, the two circulatory systems, come very close together but don't actually touch. This would, in the event that blood actually did mix, essentially the mother's body would develop antibodies, sort of like an allergy, and attack and kill the baby's tissues. So this is a strange arrangement. We have one organism trying to reside within an island within a second organism. So as strange as it sounds, you should really consider pregnancy to be a parasitic relationship. It really is. This is a unique genetic makeup. This individual has their own blood type, their own tissue type, their own immune system, and yet nature has allowed for this strange arrangement for this organism to suck the very life out of this organism. Oxygen, amino acids, fatty acids, glucose, anything that the mother puts into her body has the capacity to flow through this placenta and into the baby's body. Okay, so we've got the placenta. We've got the chorion, which is this pink layer surrounding the amnion and surrounding the fetal side of the blood supply. The key here is chorionic villus. You might remember a villus. It's a little finger-like projection. I think these look more like little trees or little mushrooms, however you want to think of it. the chorionic villus belongs to the baby and is completely surrounded by that protective outermost layer, the chorion. The maternal blood flows in through the placental sinus. So if you look here, it's sort of like the fingers of two hands coming together. We have the chorionic villus projecting upward, the placental sinus pouring down, and the chorion allowing for gaseous exchange and nutrient and waste exchange. What else do we see? Well, this is, of course, within the uterine layers. The endometrium is still here. And then deep to that, we see the myometrium. This meat here is smooth muscle, the muscular layer of the uterus. And later on, in the third trimester, when the woman goes into labor, the contractions of the myometrium will begin to propel the baby out. And obviously the baby will be a lot larger. We'll take a closer look at that in a future video. Last thing on this video is just to point out this figure here. The fetal blood circulation is unique and different from what we see in the adult. So if you look on this side first, you'll see the newborn baby's circulation, which looks very similar to what we had learned in general A&P, right? We have the right atrium and right ventricle shown in blue, bringing deoxygenated blood out, the pulmonary valve, pulmonary trunk, and the pulmonary arteries to the lungs where gaseous exchange occurs. So after the baby's born, that's how it works. Baby's breathing, baby needs the lungs. Bright red, freshly oxygenated blood comes back into the heart and then out through the aorta and out to the body. So that's a quick review of how it works in a newborn baby and in your body. But before you were born, look at this. Although the baby in the weeks leading up to labor and delivery may actually be, quote unquote, air quotes, breathing the amniotic fluid as a way of strengthening the respiratory muscles, there is no gaseous exchange happening, right? And so the circulatory system bypasses largely the circulation of the pulmonary circuit by using two bypasses, the foramen ovale and the ductus arteriosus. The foramen ovale is literally a hole in the heart in the interatrial septum, allowing for much of the blood in the right atrium to bypass the right ventricle by passing directly across to the left and into the left atrium. Not all that blood is bypassed. Some of the blood does circulate through the lungs, of course, to allow for the development of the respiratory system just prior to birth. But any blood that does go through the pulmonary circuit and then out through the left ventricle and into the aorta, look at what happens. There's a bypass. The right ventricle, there is a bypass into the aorta. So two different ways we can kind of skip over the left ventricle. The left ventricle is continually working throughout embryological development. The left ventricle, just like in the newborn and the adult, is the most powerful and important chamber of the heart. And so blood flows out the left ventricle into the aorta and then out to the rest of the body. So where does the umbilical cord tap into this system? Well, we can see here the umbilical vein shown in bright red. Remember, veins always go toward the heart, even in this strange case where we have a bright red vein emptying into the inferior vena cava. So that's why everything here is sort of shown in purple, right? The gas exchange is happening here in the placenta. And so we see the brightest and most significant difference between veins and arteries here in the placenta. The left ventricle of the baby's heart is providing for her own fetal blood pressure and circulation. And then we see here a couple of branches of the iliac arteries that form the umbilical arteries. Shown here in purple, arteries always go away from the heart, and so even though it's a long way from it, technically we are still downstream from the heart, and so the purple vessels carrying away wastes and carbon dioxide go to the placenta. Now if we flip back really quickly to this picture here, we could imagine that, again, these blue vessels, the umbilical arteries carrying carbon dioxide and waste, branch and branch and branch into thousands of these chorionic villi, and this is where carbon dioxide is spilled into the maternal sinus. So too urea, creatinine, other waste products, metabolic wastes are shunted here, and then through the maternal veins eventually into the mother's circulation, where her lungs get rid of the CO2, her kidneys and liver detoxify the blood. Following things in the opposite direction, the mother's blood supply is pushing arterial blood into the placental sinus, and this red oxygen will diffuse through the chorion into the chorionic villus, and then back to the baby's body, again through the umbilical vein. So you'll want to just spend some time practicing labeling this image. Maybe you'll make your own unlabeled image or use flashcards to try to reproduce this so that you can identify the different vessels and their colors and have a good idea of how the fetal circulation is different from the circulation of the newborn and the adult.